My name is Sophia Cochran, and my review is on whether or not antiviral therapeutics can cause mitochondrial DNA mutations as side effects, and this was done under the mentorship of Dr. Greg Shizelski. And to begin, I will be discussing antiviral therapeutic agents, specifically nucleoside analogs, and these are key treatment options for human viral pathogens. The two nucleoside analogs that I will be showing in this review are nucleoside reverse transcriptase inhibitors, NRTIs for short, and inhibitors of RNA-dependent RNA polymerases, inhibitors of RDRP for short. And NRTIs are crucial management options for human immunodeficiency virus, HIV, such as zidovudine, ZDV, and acetothymidine, AZT, and inhibitors of RNA-dependent RNA polymerases are key options for the treatment of COVID-19, such as remdesivir, RDV. And nucleoside analogs, as the name mentions, are analogs of either DNA or RNA nucleosides, and these are prodrugs, meaning that upon metabolism, they are activated into an active form, shown in figure 1b, which AZT, a thymidine analog, is converted into AZT triphosphate, which then it will be incorporated into a growing strand of DNA, where then it will inhibit the replication machinery of HIV. And as well, inhibitors of RDRP, such as remdesivir, shown in figure 2a, it's an adenosine analog, where it will also go through a series of phosphorylations into an active form, where then it will be incorporated into RNA, where then it will inhibit the RNA-dependent RNA polymerase of COVID-19. And to understand how these antiviral therapeutics can cause mitochondrial DNA mutations, we have to take a look at the mitochondria. And the mitochondria itself is the main ATP production site, and this is through the electron transport chain and oxidative phosphorylation. And not only this, the mitochondria's DNA encodes 13 oxidative phosphorylation genes, which are responsible for maintaining oxidative phosphorylation and the electron transport chain. And disruptions in mitochondrial DNA can cause serious ailments, including skeletal and cardiac myopathy, and approximately 1 in 5,000 individuals have genetic diseases caused by mitochondrial DNA mutations, which may seem like a small percentage of individuals in the population. However, approximately 1 in 200 individuals contain pathogenic mitochondrial DNA mutations, and the accumulation of these mutations can accumulate in later generations, causing mitochondrial DNA diseases. However, the cause for these mutations are unknown, and this warrants the need for research in this field. However, in this review, I speculate that nucleoside analogs have the capability of causing mitochondrial DNA mutations, and this is because the mitochondrial DNA replication system has viral ancestry, and studies have shown that nucleoside analogs can off-target the mitochondrial DNA machinery, replication machinery, shown in this figure, that AZT, it undergoes its phosphorylations into AZT triphosphate, where then it can inhibit DNA polymerase gamma, the sole replicative enzyme of mitochondrial DNA. Not only this, it can also inhibit pol gamma's proofreading mechanisms, and with these two can cause dysfunctions in mitochondrial DNA synthesis and replication, causing a decrease in oxidative phosphorylation genes expressed, which then in turn can cause disruptions in electron transport chain and oxidative phosphorylation, causing a depletion in energy. And this depletion in energy ultimately can cause damage to mitochondrial DNA. And moreover, this can be better explained by the mitochondrial dysfunction hypothesis, a three-part hypothesis that is used to explain NRT toxicity. The first step being mitochondrial DNA replication defects, such as mitochondrial DNA depletions or a decrease in DNA copy number, which then will decrease the amount of oxidative phosphorylation genes expressed, causing a disruption in electron transport chain and oxidative phosphorylation, where then there will be a decrease in ATP, which causes reactive oxygen species. And this goes into step 2, which is oxidative stress. And oxidative stress is defined as the accumulation and production of reactive oxygen species in regard to antioxidant defense, where then studies have shown that reactive oxygen species can oxidize mitochondrial DNA, which is shown in A and B. And this oxidized guanine is a mutation called the 8-oxoguanine mutation. And this leads to the study question of whether antiviral therapeutics can cause mitochondrial DNA mutations. And on to the results. Step 1, mitochondrial DNA depletions. And I have created a table of 4 references that have measured relative mitochondrial DNA content after treatment of the therapeutic agent with its respective concentrations and days under the following cell lines. And the results showed that 2 out of the 4 references showed significant decrease in mitochondrial DNA content. Reference 2 and reference 4, these 2 references, or these 2 studies being on NRTIs, and the results showed that DDC had the highest decrease in mitochondrial DNA content. Following is DDI, D14, and then ZDV. It is also important to mention that tenofovir did not show any significant decrease in mitochondrial DNA content. However, and this was also the same with RDV in reference 3 and 11, that showed no significant decrease in mitochondrial DNA content, which RDV, the inhibitor of RDRP, will no longer be assessed in this review since there was no significant mitochondrial DNA depletion. This goes on to result number 2, oxidative stress. And in a study by J. de la Ascension, they measured the levels of 8-oxo-DG, which is that oxidated guanine, in the urine of both humans and mice, and within 4 groups of humans, which we can discuss now. So in figure 1A, they measured these levels in healthy individuals, patients with HIV, and patients with HIV treated with AZT. And then lastly, patients with HIV treated with AZT and an antioxidant. And the results showed that patients treated with AZT alone had the highest levels of 8-oxo-DG. And compared to the other groups, it is also important to mention that patients with HIV treated with AZT and the antioxidant had the lowest levels of 8-oxo-DG. This was also performed on mice, which showed similar results to humans. However, within the mice study, they measured peroxide generation with fluorescence. And peroxide is a reactive oxygen species. And the results showed that in C, mice treated with AZT alone had the greatest amount of fluorescence and therefore the greatest concentration of peroxide generation, and compared to A, which was the healthy mice treated with nothing, and B, mice treated with AZT and an antioxidant. And now on to the last result of mitochondrial DNA mutations in a study by A.M. Martin et al. They identified mitochondrial DNA regions with sequence changes, and the results showed that, and this was performed in patients with HIV before and after treatment of analog reverse transcriptase therapy, and T1 represents before and T2 represents after therapy. And the results show that 5 out of the 16 patients had either non-coding or coding regions of mitochondrial DNA with sequence changes. And if you look at patient 12, patient 15, and patient 16, they had coding regions of mitochondrial DNA with sequence changes. And now on to the analysis of the results. The results are consistent with the mitochondrial dysfunction hypothesis that DNA polymerase gamma is inhibited by nucleoside analogs, which leads to a loss of mitochondrial DNA copy number, causing an energy depletion from a lower oxidative phosphorylation gene expression. And this will cause a depletion in ATP, causing reactive oxygen species to be produced in accumulation. And in turn, this will provide or cause oxidative stress. And as well, these reactive oxygen species can oxidize mitochondrial DNA into the 8-oxoguanine mutation. And then since the pol gamma repair mechanisms are inhibited, this incorporation of that mutation will, or in turn, will become replicated and not be able to be fixed by these repair mechanisms. In conclusion, the cause of mitochondrial DNA mutations are unknown. However, I speculate that nucleoside reverse transcriptase inhibitors have a potential to cause these mutations, but inhibitors of RDRP were inconclusive due to insignificance of the mitochondrial DNA depletions. The importance of this study is that pharmaceutical therapies are crucial for the management of HIV to increase longevity and quality of life. However, these mtDNA mutations may not affect the user themselves, but heritability factors can increase the risk of mitochondrial DNA mutation passage and accumulation in later generations. So the assessment of safer options are crucial, such as seen in tenofovir, where there was no significant decrease in mitochondrial DNA. However, if safer options are not available, prevention of oxidative damage is important. And this is through the use of antioxidant treatment along with NRTI treatment. As well, there's limitations to my study, including some of the references I used were 10 years or older, and few primary sources were assessed and therefore cannot accurately represent the population as a whole. As well, this is limited to the mitochondrial dysfunction hypothesis and ignores other biochemical pathways or other elements that are involved with mitochondrial DNA mutations. Thank you.